Hello friends. So today we are going to study about a very special and a unique topic called as the cytoskeletal elements. So as the name suggests, cyto and the skeleton. So as you know, the skeleton plays a major role in our body for maintaining the shape and structure of our body. In the same way, the cells present in our body also needs to have a proper shape and proper function, right? So cytoskeletal elements which are present in the cell helps in maintaining the shape of the cell as well as its organelles and in the movement of the cell from one place to another place, and so many other functions it is having. Okay, so cytoskeletal elements are of like three types. One is the microtubule and intermediate filaments, and third is actin filaments. So microtubule, let's study it in the detail. So microtubule is made up of three basic subunits. So all the filaments like microtubule, intermediate filament, and actin filaments, these are made up of small subunits which later on polymerizes and then becomes into a filament, right? So microtubule is made up of three basic subunits called as alpha. So subunits are alpha, beta and gamma subunit. So this beta and gamma subunit form a dimer, and they form the basic structure of the microtubule, whereas this alpha subunit will be helping in starting the nucleation. So wherever you find the word nucleation, it basically means that it is helping in starting the polymerization. So a microtubule is having its subunits, and these subunits have to nucleate, that is they must polymerize to form a filament, that is a microtubule. So this alpha subunit helps in starting the nucleation. Nucleation is polymerization. Okay, and the coming to the intermediate filaments, they are associated with the desmin. Second is they are associated with NF, those are the neurofilament, and next is keratin and vitamins. The actin filaments, uh, these are formed from the globular, those are the globular filaments, those are G filaments to the F actin. Sorry, it is G actin to the F actin. G actin means globular actin to fibrous actin. Okay, so actin filaments are formed from the globular actin to the fibrous actin. So the basic difference between the microtubule and the actin is microtubule uses GTP for polymerization, and here actin uses ATP for polymerization. Okay. Now we'll study some basic drugs associated with these microtubules, and those drugs are related drugs. Drugs are vincristine, vinblastine, colchicine, and Taxol. So in exam point of view, this vincristine and Taxol are very important. This Taxol is used in breast cancer in Chinese medicine. Okay, and another is colcimate. Okay, and the drugs associated with the intermediate filament are vithaferin A. Vithaferin A. The drugs associated with actin filaments is phalloidin and cytochalasin B. Okay, if you are preparing for any kind of competitive exam or something like that, so this part is like really very important to remember, because these are really asked in those exams. Okay, so now we'll see where these microtubules are found, these intermediate filaments are found, these actin filaments are found. So basically the microtubules are found in the, 1 minute, the flagella, cilia, spindle, the vesicular trafficking, it is found in forming the cell shape and the cell plate formation. Okay, and these intermediate filaments are found to form the cytoplasmic framework. And second, it is found to form the shape of organelles. When coming to the actin filaments, these are found to form the microvilli, okay, and pseudopodia, and the contractile ring. This ring is found when the cell is dividing, you find a ring here. So this ring is called as the contractile ring. Okay, so now we'll be talking about the structure of our microtubule. Basically, I told you it is made up of the beta and gamma dimer. So these are arranged in, these are hollow in shape and cylindrical. So these are like this, they're hollow and cylindrical pipe-like structure. So when you see the section, you'll find something like this, and there are 13 micro there are 13 subunits here: 1 2 3 4 5 6 7 8 9 10 11 12 13. Okay, so these are arranged in this fashion, and here I told you they are the dimers. So below this you find the another dimers present. Let me draw it in another shape. So here you find them. Yeah, yeah, here dimer. So they are arranged in this fashion. Okay, so and so these both, the alpha and the beta dimer, both form a protofilament. Okay, and these are arranged in lateral interaction. So these form the lateral interaction. Okay, so the there are two exceptions. No, I cannot say it's exception, and it's like 13 you find in major of the microtubules, but when coming to the cilia and flagella, you see a different thing. So you know cilia are small and they are many, whereas here they are present over the surface. The flagella, they are few and long, and these are found anywhere at the end. Okay, and these form the 9 plus 2 arrangement. You see, this is our section. So these are, they are 9, 2, 3, 4, 5, 6, 7, 8, 9 dimers present here. Okay, 9 dimers you found here, and the 2 you have free. There are 2 singlets here, so there are 9 doublets and 2 singlets and singlets. This is 9 plus 2 arrangement. Okay, whereas coming to the centrosome, centrosome is also a part of microfilament. So here you find 9 plus 0 arrangement. Okay, here you find 1, 2, 3, 4, 5, 6, 7, 8, 9. Here you don't find the dimer. Here you find the tetramer. Tetramer. So here this is the structure of the centromere microfilament. Sorry, the microtubule. So that's it for today. Hope you guys liked it, and if you have any doubts, you can put it in the comment section. Thank you.